Welcome to Dr. Balaga's podcast for the Curious Doc. Today is June 25th, 2026. This video is on a Nature article published yesterday, 24th June 2026, titled Dietary Cholesterol Activates a RAL-Dependent Pathway Driving LDL Receptor Turnover. There is an accompanying research briefing published today online, 25th January 2026, titled How Long-Term Dietary Cholesterol Can Slow Down Its Own Clearance by Liver Cells. Cholesterol carried by LDL drives heart disease and is cleared by liver cells expressing the LDL receptor. A cell signaling mechanism has been discovered through which high cholesterol promotes the enzyme-mediated degradation of LDL receptor. Blocking this enzyme restores LDL receptor levels in liver cells, suggesting a new strategy for treating high cholesterol. Hepatic LDL receptor governs LDL cholesterol clearance. High dietary cholesterol lowers hepatic LDL receptor, but trafficking mechanisms were unclear. The study investigators used mice with high cholesterol diets, cultured mouse, and human hepatocytes, the RAL-RAS genetic and pharmacologic perturbation, LDL receptor trafficking imaging, Cathepsin A inhibition, and human genetic analyses were utilized. What did they find? Chronic cholesterol activated the RAS-RAL signaling pathway, routed LDL receptor to lysosomes, impaired recycling, and acted independently of transcription regulation or PCSK9. The RAL gap B deletion increased plasma cholesterol, p-value equals 0.0035. The CRAS locus associated with cholesterol was involved. CTSA inhibition, that is Cathepsin A inhibition, increased hepatic LDL receptor and lowered very low LDL and LDL cholesterol by 40%. Limitations of the study: mostly preclinical mechanistic work. Cathepsin A inhibition has not been tested for hypercholesterolemia in patients, and the benefit may be limited with genetic LDL receptor loss. Key takeaways: dietary cholesterol can sabotage its own hepatic clearance by activating the RAL-dependent LDL receptor degradation. Cathepsin A is a targetable effector deserving clinical testing. What does the research briefing say? LDL cholesterol drives atherosclerotic heart disease. LDL receptor clears LDL cholesterol through hepatocytes. Yet dietary cholesterol may suppress LDL receptor beyond gene expression effects. What was the discovery? Long-term cholesterol exposure activates RAL-A, RAL-B signaling, diverting LDL receptor away from cell surface cycling towards lysosomal destruction by Cathepsin A. What is the evidence? Cathepsin A deletion preserves cell surface LDL receptor and cholesterol-exposed hepatocytes. The Cathepsin A inhibitor treatment reduced LDL, VLDL cholesterol in RAL-activated mice. The expert view: the pathway is independent of established lipid-lowering axes, making it potentially translationally important rather than another mere cholesterol footnote. The next steps: test Cathepsin A inhibition targeted strategies in hypercholesterolemia, define likely responders, and study combinations with LDL receptor raising or cholesterol absorption lowering therapies. Clinical pearls: LDL receptor abundance is not transcriptional. Chronic cholesterol stress can redirect receptor fate towards lysosomal degradation. Mechanistic pearl: RAS activates RAL. RAL engages RAL BP1 and REP-S1 pathway, suppresses SNX17 mediated LDL receptor cycling, and enables Cathepsin A mediated LDL receptor degradation. Therapeutic implication: Cathepsin A inhibition preserved LDL receptor and lowered atherogenic lipoproteins preclinically, and this may complement, not replace, statins, ezetimibe, or PCSK9 targeted therapy. Interpret cautiously, human data are genetic associations, and SARS 164653 has not been tested for cholesterol lowering. No patient outcomes are reported. Patient-facing message: some high cholesterol states may reduce the liver's ability to clear LDL. Future therapies may protect LDL receptor itself. This is an infographic of the RAL-GAP-B pathway, how dietary cholesterol promotes LDL receptor degradation. Hit pause and read at your leisure. This is an infographic on the whole article. Hit pause again and read at your leisure. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.